that this is data I'm sharing with you and is not published yet, and I would like not to be shared by other people except the people who are listening to this talk. Before, I usually forget to thank the people I collaborate with. This is the list of current scientific collaborators at the New York Blood Center. Hong Xia An is really responsible for a lot of the data I'm going to be presenting today. And then Lionel Blount Feinstein is a very close collaborator and actively involved with most of the work here. And then as part of the program project funded by NIDDK, one of the projects is at Yale University led by Pat Gallagher with assistance of Vince Schultz. And I work closely with my daughter at Stanford University, Mohandas Narla. And at Columbia University, Dr. Abdullah Ali and Razia gives us a lot of samples with myelodysplasia. I'll show you a little bit of data on that. And the proteomics data and other erythropoiesis data in Paris is done in collaboration with a fantastic biochemist, Patrick Mayo, and my former student, Lily DaCosta. And then as part of the program project, we also have a project in University of Montpellier where Naomi Taylor and Sandrina Kinnett are part of the program project as well. So the work I'm talking about is really a combination of work with very close colleagues for many years. As you know, most of you know, anemia is a major global health problem affecting at least 1 billion people. And the causes of anemia are twofold. One is increased rate of destruction of circulating red cells in disorders that include red cell membrane disorders, sickle cell disease, thalassemia, immune hemolytic anemia, and malaria. So this is the thing I've spent most of my early part of my career understanding is the mechanisms of increased red cell destruction in these different types of human red cell disorders. But during the last 10 to 15 years at the New York Blood Center, we shifted our focus to understanding erythropoiesis because decreased red cell production in the bone marrow due to ineffective erythropoiesis is also a cause of anemia. And in this case, it includes thalassemias, congenital dyserythropoiesis, and hematologic malignancies such as myelodysplasia. So today I'm mostly focusing on this decreased red cell production and both normal red cell production and disordered red cell production. As most of you realize that human erythropoiesis is a pretty complex process which occurs in the human bone marrow, starting with the hematopoietic stem cells, which once it commits itself to the erythroid lineage, we subdivide them for convenience into 3 stages. The first stage is basically generation of erythroid progenitors by the hematopoietic stem cells. And then the second stage is the terminal differentiation, where the first recognizable committed erythroid cell in the bone marrow, the proerythroblast, undergoes terminal differentiation, undergoing 5 cell divisions to generate an orthochromatic erythroblast. And this orthochromatic erythroblast at this stage will undergo what's called asymmetric cell division, generating a reticulocyte and a enucleated reticulocyte and pyrenocyte. And this reticulocyte matures over 48 to 72 hours with a mature red cell. So, we know a lot about terminal erythroid differentiation, little less about reticulocyte maturation, but really one of the black boxes in my judgment about this erythropoiesis is erythroid progenitors. And basically these erythroid progenitors have been decided mostly by functional assay by their ability to form colonies of methylcellulose cultures. And basically the 2 types that have been described are the burst-forming erythroid unit and the colony-forming erythroid units. And our guess is that there are about 8 to cell divisions within a stem cell to generate a proerythroblast. And I'll show you some data to justify this concept. So what I'm going to do basically is go through each of these different stages, the erythroid progenitors, terminal differentiation, and reticulocyte maturation in human erythropoiesis, and try to see what the kind of work we're trying to do to better understand this whole continuum from stem cells to the reticulocytes. So starting with human erythroid progenitors, as I said, what we can talk about is the BFUs and CFUs. And even earlier, it was thought that the BFU is heterogeneous just based on the colony sizes. You can either get a very large burst-forming erythroid unit, and then what's a little smaller is called intermediate, and then the smallest is obviously small BFUE. which then leads to CFE, which then gives rise to proerythroblast. And we are estimating that about 10 to cell divisions from the stem cell generation of the proerythroblast. And the cell cycle time is very fast. At this stage, anyway, our current guesstimate is that it takes about 8 to 10 hours of cell division during this erythroid progenitor stage. So a few years ago, we published a paper trying to isolate these BFUs and CFUs based on cell surface markers. And what we found was using 3 cell surface markers, IL-3 receptor, CD34, and CD36, we can actually get pretty pure populations of human BFUs and CFUs. What we found was IL-3 receptor positive cells, when you put them in methylcellulose, you get mostly myeloid colonies. Whereas if you take IL-3 receptor negative cells and put them in methylcellulose, you get erythroid colonies. So then what we did was to take the IL-3 receptor negative cells and then look at the expression of CD34 and CD36. And what we found was the CD34 positive, CD36 negative cells give predominantly BFUA colonies, whereas CD34 negative, CD36 positive cells generate CFUs. These are reasonably pure because if you plate 200 cells, we get about 150 BFU colonies, so about 75% purity of this BFU cell population based on these 2 markers. Whereas if you plate the CD34 negative, CD36 positive cells, if you put 200 cells in the methylcellulose, we get almost 160 to 170 CFUs. So we are reasonably confident that we are able to really significantly enrich for human BFU and CFE based on these 3 cell markers. Just to be sure that we are really talking about reasonably pure population, we actually did the RNA-seq analysis on these isolated cell-sorted BFUs and CFE populations. And you can see based on this, the first cluster of genes are expressed predominantly in CD34. And there's no expression at all in the erythroid lineage. Then the second set of genes are turned on at the BFU stage, but not expressed at the CFU or proerythroblast stage. And then this progresses where the last cluster basically is only expressed in proerythroblast. So at least this gives us some confidence that the populations we are isolating based on these markers are reasonably specific, and they do in fact represent BFUs and CFUs. But as I said earlier, that even when we take the so-called purified BFU populations and put them in methylcellulose cultures, we either see very huge colonies of BFU, which we call large BFUs, or some which are smaller than BFU, but a single big cluster, which is intermediate BFU, and then the small BFUs. Whereas the CFU colonies we isolate are basically pretty uniform, and we don't have as much diversity as you see in the BFU colonies. So the challenge was maybe we need additional markers besides CD36, IL-3 receptor, and CD34 and 36, and IL-3 receptor negative to get more discrimination on these BFU subpopulations. So the last number of years, this is some unpublished data, we are writing up this paper, is basically based on the analysis of RNA-seq, we found that the 2 potential candidates to do a better job is CD71, the transferrin receptor, and CD105, endoglin, which is the TGF-beta superfamily of proteins. And when we now combine this CD71 and CD105 along with CD36 and CD34, we now are able to get at least 5 different populations of erythroid progenitors. The CD71 negative population, the P1 here, basically gives rise to large burst-forming units, but also some myeloid colonies. So we have a mixture of both myeloid and erythroid. But once we choose CD71 expressing BFUEs, then you can see we get very large immature BFUEs. And then as the CD105 expression goes up, like P4 and P5, you can see the colony size goes down, and the P5 population is only mature CFUs. So at least just from BFU to CFE, now we are able to at least get 5 distinct populations. And I must say there's still more to be done in terms of it's likely that we can find additional markers to get better discrimination than even these 5 subsets. And working with Pat and Vince, we're able to show that when we do RNA-seq analysis on that, this is the original RNA-seq analysis based on CD34 and CD36, and you can see the BFUs are here and CFUs are here. But now with the new populations of EP1, EP2, EP3, and EP4, you can see we get a much broader spectrum of separation between these populations, whereas clearly the proerythroblasts are all very, very distinct populations. So now we feel confident that we can, in fact, identify at least 4 distinct populations of human erythroid progenitors based on these assays. So what I've showed you so far is that the heterogeneity of progenitor populations appears to relate to the number of cell divisions of CD34 cells, and surface markers CD34, CD36, CD71, and CD105 enable the isolation of several distinct subpopulations of erythroid progenitors. What we're doing now as part of the program project, we're doing gene expression, epigenetic, metabolomic, and proteomic studies on these purified progenitor populations to obtain further insights into human erythropoiesis. Now, switching on to human terminal erythroid differentiation, as I said earlier, this has been very well studied for a number of years, and in fact has been shown for a long time. The proerythroblast undergoes 4 to 5 cell divisions to generate orthochromatic erythroblasts, which then enucleates to ureticulocytes and pyranocytes. And interestingly, in contrast to what I told you about erythroid progenitors, where we think the cell division takes place every 8 to 10 hours, in this case, the cell cycle is much slower. It takes about 20 to 24 hours for the cells to divide. And this has been well documented for a number of years. Again, a number of years ago, we tried to see if we can purify from human terminally differentiating erythroblasts all the 5 distinct populations of terminally differentiating erythroblasts. And in this case, what we found was by using 2 glycophorin A, alpha-4 integrin, and band 3, we in fact can separate distinct populations based on these 2 markers. The alpha-4 high integrin, low band 3 correspond to the proerythroblast population, and further increase in band 3 expression gives rise to early baso, and then late baso, and then poly and orthochromatic. And we get about 90 to 95% pure, at least consistently, of these 5 distinct populations based on these 3 markers. Again, with the help of Pat and Wells, we actually did the RNA-seq analysis, and again, it gives you a very clear idea. This is 3 biological replicates. You can see the proerythroblasts have a distinct RNA-seq expression pattern, and then as you go to later and later, you get increased expression and decreased expression of various sets of genes. Again, this is really to show that what we are purifying are really distinct populations, and based on morphology, as I showed you, there really are proerythroblasts, early basal, late basal, and poly, and orthochromatic erythroblasts. But I always was a little concerned that RNA-seq gives us the messages and all, but what we really care about is the amount of protein being made at these different stages. So this work was done by Emily Flair-Gauthier in Patrick Mayou's lab. And what they did was to basically get quantitative analysis of the amount of proteins expressed at these distinct developmental stages. And so what they were able to identify, about 7,300 proteins were identified. And of these, about 6,160 proteins could be quantified with copy numbers per cell. How this is done is basically, how do you get the copy number is basically all these cells, as you can imagine, have a 2N DNA content. So they use the histone peptides as the denominator on the tryptic maps. And so you can actually use that as a denominator and really get exactly the number of copy number of the proteins. For example, you can see, as you can expect, The hemoglobin is the biggest component. You have 10 to the power 8 to 10 to the power 9 molecules per cell. Whereas as you go down the list, if you look at MIB and MIT, you can see there are about 1,000 copies or 1,500 copies of this cell. So we really have a very good understanding of at least the 6,000 proteins that we are able to quantify at these 5 distinct stages. What I need to mention though is that when we have quantification, that we are very confident, but it doesn't mean we are identifying all the proteins for 2 reasons. One, if they're below a certain copy number, we may not have the detection sensitivity to detect them. The second thing is the tryptic peptide that's generated by that protein is not in the range of the sizes that is detected by the mass spec. We also won't be able to. So this is a minimum estimate we can get, but there could be more proteins than what is shown here. There are some of these published protein copy numbers previously done by other investigators, and you can see when we compare the data generated by this technology, you can see a pretty nice relationship in the copy number previously published and what we determined here over at least a 5 to 6-fold log range of proteins. So we're pretty comfortable that this proteomic analysis will be useful to understand erythropoiesis. So the question is, I showed you RNA-seq data and the proteome, and the question is, what is the relationship between the transcriptome and proteome? And you can see it's reasonably good between the two systems, except of the 300 identified genes with significant divergence between protein and mRNA expression. So in this case, we have, for example, if you look at SF3B mRNA, you can see it's going up as the cells are terminally differentiating, but the protein content is going down. So really, there's not a direct relationship between the two, at least for these about 300 identified proteins. And what was interesting to us, because John Conboy, who I worked with many, many years in Berkeley, was very interested in alternative splicing and splicing mechanisms. And at least in the 53 of these 300 identified genes where there was a divergence, there was intron retention state specific. So by retaining an intron, although your messages can be translated and you can make the protein. This intron retention is becoming a very interesting area in terms of gene regulation and protein expression. And we just started, really very early stages of understanding what this process is during human erythropoiesis. And as part of our program, actually, Pat is the project really doing a great job of epigenomic landscape of human erythropoiesis. And recently, Vince and Pat had a very nice paper actually on the epigenomic landscape that defines human erythropoiesis. And people who are interested should really have a good look at this paper. And again, we are continuing these studies and getting more and more depth into really understanding regulation at the epigenomic level during erythroid differentiation. Basically, in terms of the present state of the thing, we are continuing this RNA-seq as well as this thing. And we are in the process of generating proteomic data, all the 4 different erythroid progenitors I talked about, EP1, EP2, EP3, EP4. And the experiments are currently being done, and we are hoping we should be able to have the proteomic data in the next few months. The other part of our program is really metabolic regulation of erythropoiesis, and this is the project led by Naomi Taylor and Sandrina Kenneth as part of our program project. And this is what started the whole thing. I got interested and started working with them, is because a few years ago, Naomi Taylor's group showed that glucose and glutamine metabolism regulate hematopoietic stem cell lineage. And in fact, this regulates the commitment either to erythroid lineage or myeloid lineage, depending on the balance of glucose to glutamine metabolism in these stem cells. So what we are trying to do now is, since erythroid differentiation is associated with increased metabolic requirements, and increased metabolism causes cells to adapt to facilitate uptake of nutrients by regulating surface expression of nutrient transporters. And what we are trying to do is we want to define complete, as good as we can, all the different nutrient transporters that are expressed during erythroid differentiation. And the way we are able to do that is a number of years ago, Mark Sidbone and Naomi Taylor in Montpellier found that the retroviral envelope receptor binding domains, that is, the various retrovirals that have the receptor binding domain, actually bind specifically to various nutrient transport proteins expressed on cells. For example, HTLV1 receptor binding domain binds specifically to glucose transport. And since then, they have identified a number of other RBDs that bind to specific nutrient transporters. And the way we do these experiments is we take the RBD binding domain and make a recombinant RBD binding domain for various viruses, and then tag it with a FC portion or a fluorescent protein. And this is not like antibodies, they just bind directly the receptor with high affinity to membrane transporters, so we can actually do intact cells and see how much fluorescence or antibodies are associated with the cells at different stages. So we've been able to actually study at least 9 different transporters: GLUT1, phosphate importer, and phosphate exporters, and heme exporters, and riboflavin importers, and arginine importers. So basically, these are the different viruses, and we are able to actually synthesize the RBD domains from these 9 different viruses and actually use them to study expression on erythroblasts at different stages. So just to give you an example, as I told you, we are able to use these markers to separate both the progenitors as well as the terminally differentiating erythroblasts. And using, say, the GLUT1 RBD, we can say is lowly expressed at IL-3 myeloid cells, but start to get more expressed at BFE, and continue GLUT1 expression goes up to all the way to erythroid progenitor development, and is also present at high levels in proerythroblasts and continues to be expressed through erythroid differentiation. So we really are able to map exactly the number expression pattern of this transporter throughout from BFE all the way to the reticulocyte. Using these 9 RBD domains, I talked about riboflavin importer, phosphate importers, phosphate exporters, heme exporters, and all. Now we have a pretty good mapping of all these different 9 transporter expression, all the way from HSC to orthochromatic erythroblast. And you can very clearly see that pattern is very different. For example, the glucose transporter is expressed very highly, whereas the other transporters are expressed at early stages of progenitors, but then go down dramatically during terminal erythroid differentiation. So this is a good data set for us to have. We have a lot of work to do, but at least we know these are the proteins, that transporters that are expressed. And now the question is, what is the functionality of these different transporters during erythroid differentiation? That's what we would like to study in the coming years. So what this shows is that receptor binding domain have enabled us to evaluate changes in expression of nutrient transporters throughout human erythropoiesis, and all transporters showed varying levels of expression through various stages of human erythroid differentiation, with each stage exhibiting a unique metabolic signature. And each stage of human erythropoiesis has a unique metabolic signature. Now, what we are trying to do, as you can expect, is you're using CRISPR-Cas9 to evaluate the role of different metabolic transporters at different development stages during human erythropoiesis. It will take us a while to do that, but I feel we will be able to generate some interesting insights using this approach. So the last stage of human erythropoiesis, as I said, is when orthochromatic erythroblast actually enucleates and gives to the enucleated reticulocyte, and what my friend Jim Pallas called perinocyte, which is the extruded nuclei surrounded by a plasma membrane. And these are, you don't see them at all because they're rapidly phagocytosed by the macrophages in the erythroblastic islands. And this process really, the enucleation process itself, once it starts in the process, it takes about 8 to 10 minutes to finish the process, where it generates the reticulocyte and the perinocyte. A number of years ago, when I was in Paris with Marcel Bessis, we actually were interested in this enucleation process. So you can see this is a phase contrast image of nucleus and the enucleating erythroblast at the stage of enucleation. And then once its enucleation is finished, people don't realize the immature reticulocyte that's generated right after enucleation has a very unique morphology with multilobular. You can easily detect it by phase contrast microscopy or optical microscopy. And then it becomes a more discoid, not really, but a shallow center, and then basically become a nice discoid mature red cells. And if you look at the EM, actually, as you can see in this immature reticulocytes, there are a lot of ribosomes, a lot of mitochondria, and it's very distinguishing feature of these cells. Whereas once you go to the later stages of reticulocyte maturation, the ribosomes are degraded, mitochondria are being modified, and you get total elimination of the mitochondria by the time it becomes a mature red cell. So this is, to me, this is a very interesting problem because this happens very rapidly and it's very selective, right? I mean, a lot of proteins that are destroyed, like for example, the transferrin receptor, ribosomal protein, mitophagy and all, are very specific, and this happens very rapidly following enucleation. My guess is this process takes only about 24 hours, and a lot of interesting biology to study during the reticulocyte maturation process. So what we have been trying to do again as part of this program is try to understand more about human reticulocyte biology. And what we would like to do is, again, using the same strategies of surface markers and cell sorting to be able to isolate different stages of reticulocytes. For example, if you look at CD36, it's highly expressed on the orthochromatic erythroblasts and is completely lost by the time they become mature red cells. And if you look at the reticulocyte, you can get a spectrum of CD36 expression. So if you plot CD36 against CD70, again, we get this very nice unique pattern. And the question is, can we isolate these different stages and do the understand this maturation of the immature reticulocyte into the mature red cell? That's what we're trying to do right now. And during this process, not only CD36 changes, but CD71 is highly expressed on retics, but as I said, is completely lost by the become mature red cells. Same thing with beta-1 integrin and alpha-4 integrin. So really, we have a number of markers we know are changing during reticulocyte maturation, and we're trying to optimize the best combination of markers that will give us all the different stages of reticulocyte maturation into mature red cell. We just started again as originally a small project with Patrick Mayu's group, where we try to purify reticulocyte from peripheral blood, because as you know, the peripheral blood has only 1% or so reticulocytes in normal people, 1 to 1.5%. So we, by self-sorting, we're able to identify almost 99% pure circulating mature reticulocytes, and then compare them with mature red cells for proteomic analysis. What I want to point out, these cells in circulation in adults is really very mature stages, and this corresponds to the very late stages in reticulocyte biology. And again, what we're able to do is do the quantitation of proteins. In this case, in contrast to using histone because of DNA, we are using globin because each red cell and the reticulocyte both have about 30 picograms of hemoglobin per cell. So we can use the globin peptides as the denominator to quantify proteins in intact red cells or intact reticulocytes. And we quantitate about 1,000 proteins in the mature red cell. And in these mature reticulocytes, we have about 350 to 400 more proteins in the reticulocyte compared to the mature red cells. And I'm pretty confident when we do the quantitation in the very immature reticulocyte, we'll have at least 3,000 to 4,000 proteins, and that's what we're trying to currently establish. So the last part of the reticulocyte is really using these different cell surface markers. We hope to isolate reticulocytes at different stages of maturation from human bone marrow. And that is the trick, actually, because in peripheral blood, you rarely see these immature reticulocytes. You would not be able to isolate from peripheral blood. So what we are doing now is, because we are able to get human bone marrow, that's where all these cells are maturing, and we are able to show that we can get, in fact, all different stages of reticulocyte maturation by working with, not with peripheral blood, but with human bone marrow. And we'll use the state-of-the-art proteomic studies to quantitate protein composition of reticulocyte at different states of maturation. And we are using some novel biophysical strategies to determine the structural organization, because as I said, the immature reticulocyte is motile, there's a lot of surface motility, there's a lot of myosin and tubulin, also proteins that are not present in the red cell. So we expect them to have unique biophysical characteristics compared to mature red cells. And why we want to do that is I strongly believe that defects in reticulocyte maturation is likely to be a feature of red cell disorders such as membrane disorders and sickle cell disease. And it'll be nice to know, because most people, when they compare sickle blood to normal blood, they really are not comparing fairly, because there's so many reticulocytes in sickle cell blood that really you have to account for what the measurements you're making that's due to reticulocytes and what is due to mature red cells. Finally, I told you all these things we can do in culture and in from bone marrow and all, but the problem is that the erythroid differentiation in the bone marrow occurs not in isolation, but in three-dimensional islands called erythroblastic islands. So basically, this is where all the erythropoiesis occurs in human bone marrow in the context of erythroblastic islands. So it consists of a central macrophage surrounded by developing erythroblasts. And it's been described almost 60 years ago by Marcel Desis and Madame Rotangurius about the importance of erythroblastic island. And it didn't really receive much attention for many, many years. Now I think there's a lot of interest by a number of different groups on the role of macrophages in erythropoiesis. And what we can be sure of is that what is the function of the macrophage in the erythroblastic island? Clearly, it phagocytosis of the extruded nuclei or the peronocyte, because when you take the bone marrow, you rarely see any of these peronocytes, even in the aspirates, whereas we can see a lot of them in cultures where there are no macrophages. So clearly, this is important to remove the nuclei as soon as they're extruded. And then there is some data from Mark Curry and others that also is involved in regulation of erythroblast proliferation. And more, there's a lot of interest, especially for people interested in iron homeostasis, that macrophages play an important role in heme biosynthesis and transferring ferritin and heme and other things from the macrophage into the erythroblast. We are not working on it, but it's a very interesting, a lot of exciting work is coming from other groups about the role of macrophage in iron homeostasis in human erythropoiesis. As I said, erythroid differentiation occurs in the context of erythroid niche consisting of terminal differentiating erythroblast in intimate contact with the central macrophages. And macrophages play a critical role in regulating terminal erythroid differentiation by regulation of cell proliferation, iron supply, and in rapid removal of extruded nuclei. My hope is, using these highly enriched populations of human erythroid cells at distinct stages of development, and our macrophages directly isolated from intact bone marrow erythroblastic island, it may be possible to begin to decide for the role of erythroid niche in regulating normal and disordered terminal erythroid differentiation. So in summary then, I hope I showed you that the ability to quantify and purify erythroid cells at all distinct stages will enable us to define stage-specific defects of erythropoiesis in various human diseases. Ineffective erythropoiesis and thalassemia we have already shown is due to defects at late stages of terminal erythroid differentiation. And I didn't have time to show, but we also showed using the methodologies I described to you that ineffective erythropoiesis in diamond black-fang anemia is primarily due to defective development of erythroid progenitors, while the ineffective erythropoiesis in adults with myelodysplasia is due to defects in both erythroid progenitor development as well as in terminal erythroid differentiation. And I'm hoping using this highly enriched population of human erythroid cells at distinct stages of development and of macrophages isolated directly from erythroblastic island will also start to decipher the role of erythroid niche. And so I'm still, after all these years, still very excited about working on red cells and erythropoiesis. And I think there's still a lot to learn. And I think all the new technologies that are coming on board using single cell sequencing and single cell epigenetic studies and all, I think we'll continue to make interesting progress in a detailed understanding of human erythropoiesis and also applying it to study ineffective erythropoiesis in various human red cell disorders, including thalassemia, MDS. And there's some recent interesting work from my colleague in France, where we were also able to demonstrate ineffective erythropoiesis in sickle cell disease, which was not that much appreciated in previous years. So thank you very much, and I'm happy to answer any questions.
Wonderful. Thank you very much, Dr. Narla, for your talk today. You're welcome to unmute yourself and ask specific questions at this time. Kind of just giving people a minute to do that.
Okay. I'll stop sharing, okay?
You can, unless somebody wants to ask a question specific.
No, I'll be fine. No, it's fine.
Yeah.
I have a question, Mohan.
Sure.
Have you looked at the secretome of erythroblasts? Because there must be, although we have some initial clues about what they might be secreting, we don't have a comprehensive understanding of that. Have you looked at that?
No, not at all. That's a good point. Actually, now that we have the proteome of these different stages and all, and also, I guess we need to do that in the context of serum-free media, though, right? I mean, otherwise that would be too much. When we put 10% plasma or fetal cap serum and all that kind of stuff, is really a problem. So it'll be something worth thinking about. Actually, we could be able to do that, taking supernatant from these cultures and looking at what is being secreted there.
Because it's likely that there are multiple hooks into systemic physiology by the developing erythron, and it would be nice to characterize those.
Sure, that's a good point. I see Dr. Sam Lux is here. He's the man who got me started in the red cell field. He gave me my first grant 45 years ago.
Hi, Sam.
Other questions?
This is Jen Apkowitz. I have a question. First of all, great. It was super to have you put all this together. I wanted to go back to your statements about cell cycle, and the question is how different ways have you calculated that? Is that in vitro? Is it in vivo? Do they differ? And a corollary of that is, if you had stress erythropoiesis or erythropoietin or something of that sort, does it ever change cell cycle or just cell survival?
Yeah, that's a good point. Yeah. All I'll tell you is with cell cycle studies, what we have done is take EP1, all these different populations, and look cell trace to look at cell divisions, okay? Plus, not only that's by itself is okay, but not complete. So we are also doing BRDU and stuff. And we can clearly show that in the early stages you have, for example, like EP1 or something like that, is very slow. 30%, S phase, but as they go further and further, it's becoming much higher. We just finishing up that cell cycle data from using BRDU on these different isolated subpopulations here. And in terms of erythropoietin, that's a very interesting point, because what you raise is interesting to me, because as you know, in these cultures, we are overloading them with EPO. Okay, I mean, sometimes people use 3 units of EPO in the cell cultures and all. So one of the things I'm particularly interested in is basically now that we're able to isolate what is the EPO sensitivity of these different populations for proliferation. So that's one of the projects we are doing right now, is EPO sensitivity of these different populations.
I think that's a super project. There's a lot of feeling, as you're probably aware, that EPO's predominant effect is on survival rather than other cells. And you're in a wonderful position to be able to decipher that.
Sure. Actually, once when we started is using a lot of EPO, then what we found is at late stages, once they go past the basophilic stage, you don't need EPO. You don't need, you just need trace amounts or no EPO. They still go and continue to, except survival, they die. They go apoptosis, but you don't need them for a lot of it at this stage.
Great. Other questions?
So I have a question. So Dr. Narla, great presentation. Hey, when your proteomic approach was very interesting. And because you also have, thalassemia and all of these other sort of underlying red cell problems at hand, have you looked at the proteomic profile in different disease states?
This is very interesting, obviously the right thing to do, but at this stage, we have focused totally on normal erythropoiesis to get the really foundational data sets for all these different proteomics and epigenetics and all these kinds of things. But clearly, once we are confident, now the next stage would be to take, for example, thalassemia at different stages of maturation and compare one-to-one at the pro-erythroblask stage or at late stages. What is the proteome in these two things? Because with all these excess globin chains and ROS and all those things, does it also affect protein expression? That's something we need to do, but we are not yet started.
Well, when you're ready to expand it out into the very important diseases of heme biosynthesis, you let me know.
Sure. You're the expert. I won't touch it. My philosophy in science is really, I don't want to get into things that I'm not confident or good at, but let other people who are really good at it do those kinds of stuff. One thing I'm quite interested, I must say, as part of the program is because our colleagues in Colombia, they have a lot of bone marrow from MDS patients. So we are getting we can get a couple of bone marrows from MDS every week if I want, or more if I can. But the reality is I'm most interested is there we have very good evidence in some of these MDS patients. The proliferation defect is really at the erythroid progenitor stage, and they really don't get any terminally differentiating erythroblasts in the bone marrow. Whereas in other cases, they have progenitors, but they're apoptosing at the later stages, and what the differences are is not very clear. That's what we would like to do, is isolate cells and see what the proteomics and RNA-seq data at these different stages and these different diseases are. Yeah.
Stefano.
Hi, thank you. Hi, Mohan. Very nice talk as usual. I'm curious. I want to pick your brain about a paper that was published in Blood last October about how the red cells basically release their heme, which is not uptaken mostly by macrophages, but mostly by proteins that recognize this molecule. And so from that perspective, macrophages seems to recycle mostly the ghost red cells based on that manuscript. I mean, it was a manuscript by Clive Bruggen, I think, in the Netherlands. And so assuming that this is obviously strong data and it's a true mechanism, etc., how would you reconsider or consider the relationship between macrophages and red cells? I mean, is that more related to nurturing them than really recycling the heme? I mean, what's your opinion?
Again, this is opinion. I don't have a lot of data, but actually, when I first went to Paris in the early 70s, Bissis was very much interested in the erythroblastic islands, and he always said, that there is exchange of ferritin for the macrophage to the erythroblast. He published some, strictly based on EM studies. It's called rophiocitosis or some terminology for that. But now, as you know, some of the people working in field in iron and heme and all these kinds of things, there's a lot of interesting transporters, right? Heme exporters like Janice has done and all those people. So it's very plausible, for me to assume there's a lot of iron that's being recycled back from the macrophages into the erythroblast. How to do that, I think, is a little bit challenging, but I think it's something very important because I think it's just, I don't think evolutionary is going to waste anything, in human biology. So I think, why would it not use what it has digested and destroyed back into the thing? And in this context, I recently heard from a good colleague that, this is again strictly speculation. They said we always look at iron in the liver and heart and all those kinds of things, And the question is, with so many red cells being destroyed in the spleen, what is happening to all that iron? And they were telling me that in some of the patients, they are seeing iron in the spleen, And whether it is due to increased destruction or not is not very clear. And at least they claim that whenever they see iron in the spleen, number of patients have hereditary spherocytosis, which Sam is interested in. So I think whether there's cycling of iron and how it is stored when you're this 5 to 10-fold increased rate of destruction of the spleen is something to be keep our eyes and ears open, I think.
So let me follow up on that question, Mohan.
Here, he's the expert.
So I'm deeply interested in erythroblastic islands, as you and Jim know, right? So I wanted to know, how does a nurse macrophage differentiate specifically in the marrow from a recycling macrophage? Does it ever decide to abort erythropoiesis and start engulfing red cells? Because in our knockout, right, in the HRGO knockout, you see hemazoin not only in the red pulp macrophage and Kupffer cells, but specific regions in the marrow. That would mean that either these are purely recycling macrophages that's recycling in the marrow, or that nurse macrophages have now decided that there's ineffective erythropoiesis going on, which is occurring in the knockout mouse, and it's now decided to also take in a partial recycling function.
No, to be honest with you, I've been thinking about it for many, many years, the erythroblastic islands, but macrophage is still a mystery to me, frankly. Yes, I think there is a lot of biology there. And I think the question you ask is very important, is the subsets of macrophages that are clearing and subsets of macrophages. I think that will be a very important issue. And all I can tell you is I think people are making, not me, but other people are making some good progress, where the way to study this problem, in my judgment, is to isolate the macrophages directly from the erythroblastic island.
I would suggest that, no, we should do this directly in the marrow.
Right, sure.
Using the right
better, even
conditions of
better.
the marrow, right? Which will be
Sure.
like 2% oxygen, not
Sure.
the oxygen that we use in our incubator. And then actually study what's happening on the
No, that-
EBI at the interface.
I think maybe these days with the new technology, you'll be able to do that,
We should be able to.
stuff like that, for sure. But the reality is, though, that I think in the old days, we were all fooled because we thought macrophages are macrophages, but not paying attention to the specificity of these macrophage subpopulations and stuff like that. But I really know very little about macrophages.
So what I was trying to say is, as soon as you pull out the EBI, it's no longer functioning like an EBI.
Sure.
That's what I'm
plausible.
going to get.
It's plausible.
Okay.
I have a question, and I'm gonna stick with this whole macrophage theme. Would your proteomics approach be appropriate for assessing whether there's direct exchange of proteins between the macrophage and the developing erythroblasts? Because we know there are structures that make it look like these cell types are communicating directly with each other, the cytoplasmic extensions.
Sure.
So could your proteomics approach be adapted to answer that question?
I'm too old to do those kinds of experiments.
Not an excuse. We are not accepting that.
But I really think that, to me, is really a critical question, because if you look at we're talking about intact bone marrow. In the old days, if you look at sections of marrow and look at the erythroblastic islands, you're absolutely right. These are not spheroidal structures. They're really big element of macrophage putting out this incredible length over many microns, and they're interacting. So that's a very interesting question. I'm sorry, I can't give you an intelligent answer to that.
Thank you.
So Mohan,
You should do that, Carolyn. If you want to do it, I will give you all the reagents to separate these things.
so Mohan,
Yes, sir.
can you hear me? Yeah.
Yes, yes.
Where are all the diseases of this process? I keep thinking that there should be diseases of late erythroblast maturation that would present as black fan diamond anemia, diamond black fan anemia in a way, but they'd be different in the sense that they'd be having a bone marrow full of erythroblasts. Maybe you'd have cells that don't properly enucleate. You can just imagine a whole series of diseases that ought to occur from all the processes you're describing. Yet I don't know of any examples of such diseases that have been described.
Right. I don't know, Sam, you can correct me, but my feeling is unless we have methodologies to study these things, right, it's very hard, right? I mean, if you look at the peripheral blood, what you're seeing is what's coming out from the bone marrow into the peripheral blood, right? So if there are defects, in the bone marrow, unless you really look, and actually, to me, what I've been told by people like you and even my Anu, is that nowadays people don't do a lot of bone marrows. In
No, they don't
fact,
do enough.
they're not even doing bone marrows on DBA patients for diagnosis because they say you can do the genetic screening and the RPR ribosomal mutation. So I genuinely think in the old days, at least I'm very biased. I think when we take an aspirate and do the cytospin and look at the morphology, you can see the growth. But I think it's much more subtle than that. And that's one of my hopes is the methodologies we are developing now. We can actually quantitate, actually, we're just finishing up a paper where I can show you all these different populations, what is the quantitatively, how many are there in human bone marrow, and then hopefully, and it may not answer what exactly we wanted, but maybe we'll see that there are more subtle defects that we have missed over the years here.
John, do you want to ask your question or do you want me to read it?
So yeah, I put in the chat. So, kind of
I'll
along
go back and answer the chat questions later if you don't mind. Go ahead, but please ask.
So along the line of Iqbal's question, what percentage of red cells in a normal bone marrow never fully develop and essentially are aborted and need to be recycled in the bone marrow itself?
1 in 10.
I can't give you a perfect answer, but my bias is in normal erythropoiesis, there's very little wastage. Really. I'd be very dubious that they're going through this whole differentiation process. And I think they'll complete the maturation, they get released into circulation. What Sam said, maybe in diseases, there is a problem. And somehow the reticulocyte is different and the macrophage says, you're not normal and I'll eat you up before. Whereas I don't think there's any evidence that normal erythropoiesis, there is phagocytosis of red cells in the bone marrow.
Why think about wastage? Why can't we just say salvage?
Sorry?
Why can't we just say salvage instead of wastage?
Yeah, but the reality is, though, it's a very finely tuned process, right? You're making 2 million red cells a second, and you don't, but clearly, if you stress the system or something, things may change. But I don't think that in normal erythropoiesis, you have a lot of waste. I think my bias is really in the progenitors. I really think there is a certain population of progenitors that if they don't mature, they will salvage because you don't need them. And the reason you have to have that kind of population in the bone marrow is when you acutely need to make red cells. Okay, like somebody bleeds a lot, lost a lot of blood, and you get reticulocyte response within a few days, right? You can get reticulocytes in 5 days, 4 days, unless you had a reserve population of cells that are ready to make red cells, but normally are not used. And this has been postulated by great hematologists in the past, like I'm blocking on his name, in Boston, and other people who really were very, I think even in Seattle with Janice and other people, I think there must be a reserve population of progenitors that ready to make red cells. And you don't want to start all the way from stem cells and go through 4 or 5 days of development to generate a pluriblastoblast. But again, there's no proof, it's a concept, and I think it's worth testing.
Good. I want to thank everybody there for their participation in today's talk, and especially thank Mohan for his wonderful presentation. Leaves a lot of thought-provoking questions and a lot of stirring to help us try and think of ways that we can actually answer these difficult questions about, are we seeing artificial systems when we're culturing in vitro that don't necessarily reflect what's happening in vivo? Really important, really, really important. Thanks
if you don't
again.
mind, I'll tell you one thing, if you don't mind,
Please.
before is that I think that's a very important point also. We need to, or when we do all this in culture, so what we are trying to do as part of our program is really validate all the data, the proteomics data, the RNA-seq data, all not only from cells from cultures of different populations, but isolate directly from the primary human bone marrow, and actually validate how concordance is there between what we do in cultures as with in the bone marrow. And I think that is absolutely a key point in all this data we're generating, I think.
Great. If you want to share your video or whatever you want, it's so nice to see everyone. And it was a great presentation, Mohan. Thank you so much.
Thank you so much. Thank you for listening. And if you have any questions, let me know. And also any good ideas, let me know too.
These are wonderful opportunities to share ideas and collaborate. So thanks everybody for your participation. And we look forward to seeing you in the next seminar, the first Wednesday of April.
Thank you, Mohan. Thank you, Dan.
Bye-bye. Thank you, guys. Thanks a lot. It's a great pleasure to see all the friendly faces in the red cell field. Thank you. Thank you.
Thank you, Mohan.
Thank you, Sam.